Welcome to the Deep Dive. Ready to explore some incredible breakthroughs in genomics and bioinformatics? We've got some really cool articles about RNA sequencing, AI and drug development, you name it.
It's amazing to see how quickly things are moving forward in this field. We're going way beyond just reading DNA now. It's like we're starting to understand the actual language of life.
Absolutely. Let's talk about this new UDA-seq technique. It's called Universal Droplet Microfluidics-Based Combinatorial Indexing. I know that's a mouthful. Basically, it's this amazing way to do massive single-cell sequencing, multimodal too.
Imagine analyzing millions of cells all at once, and not just their DNA, but their RNA and protein markers too. It's like getting a super high-def picture of a whole ecosystem, seeing how everything interacts.
So instead of a blurry image of a forest, we can zoom in on each tree and see its connections.
Exactly. Think about mapping out a developing brain, cell by cell, with all its RNA activity. UDA-seq could unlock the secrets of how our brains form, maybe even lead to treatments for developmental disorders.
That's incredible. A molecular Google Maps for the body. And speaking of understanding complex systems, there's this cool computational tool called MIT-hires. It lets scientists figure out single-cell and spatial microRNA activity from transcriptomics data.
MicroRNAs are like these tiny regulators, fine-tuning gene expression by controlling protein production. So your DNA is like a huge library of blueprints for your body. Genes are the individual blueprints, and microRNAs are the librarians, deciding which ones get used.
They make sure the right genes are on and off at the right times and in the right cells.
Precisely. MIT-hires lets us see how these microRNA librarians are working across different cell types and within tissues. This helps us understand how cells function and how diseases develop. For example, researchers could use it to study how microRNAs go wrong in cancer cells, leading to uncontrolled growth. This could help develop targeted therapies that fix those imbalances.
Fascinating. RNA sequencing is about more than just the genetic code. It's about understanding the whole molecular symphony of life. How is this tech being used to fight specific diseases?
Well, one study used RNA sequencing to investigate HIV-1, the virus that causes AIDS. Researchers at the Helmholtz Institute discovered a sneaky way HIV-1 survives inside our cells.
HIV is a tricky virus, always evolving. What did they find out?
They found out that HIV-1 manipulates our cells to prioritize the production of its own viral proteins.
Like a hostile takeover of our cellular factories, forcing them to build weapons for the enemy?
Exactly. This is a big deal because it shows us a new weakness in HIV-1 that we can try to exploit. By understanding this manipulation, we can develop drugs to block it and stop the virus from replicating. This could mean better treatments with fewer side effects.
That's good news for people living with HIV. RNA sequencing is powerful. It's uncovering the secrets of viruses and leading to new treatments. But let's switch gears for a moment and talk about another amazing use of RNA sequencing. Understanding and potentially treating a terrible neurodegenerative disease. Huntington's disease.
Huntington's disease is genetic. It causes progressive brain cell death, leading to problems with movement, cognition, and even psychiatric issues. Right now, there's no cure. But researchers at the Braun Institute are using RNA sequencing to try to unravel the disease's mechanisms.
What have they discovered?
Something really surprising. The mutation that causes Huntington's doesn't actually become toxic until later in life. That was unexpected. You'd think a harmful mutation would have immediate effects.
Like a time bomb in our genes waiting to go off. That's scary, but maybe also a good thing.
Exactly. If we can understand why it becomes toxic later, maybe we can develop therapies that target that late-onset toxicity, slow down, or maybe even stop the progression. It gives hope that Huntington's might not be a death sentence, but a manageable condition.
RNA sequencing is giving us incredible insights into so many diseases, from viruses to genetic disorders. And it's not just about understanding them, it's about personalized treatment too. I was really interested in this article about a partnership between Cipher Medicine and OMNY Health, two companies at the forefront of precision medicine.
Precision medicine is all about tailoring treatments to each patient, looking at their genes, lifestyle, and everything else.
Right. What's cool about this partnership is that they're taking genomic data from Cipher's PRISM-RA test and integrating it into OMNY's giant electronic medical record network.
The goal is to change how we treat immunological diseases like rheumatoid arthritis. Imagine a huge database with patient info, including genetic profiles and how they responded to different treatments.
That's a treasure trove of information for researchers. What can they do with all that data?
By using AI and network biology solutions, they can find patterns and predict which treatments will work best for individual patients. This could make healthcare more precise and effective. No more trial and error, just better outcomes, like a personalized medical roadmap for every patient. Cipher Medicine and OMNY Health working together is really a perfect example of how powerful it can be when you combine big data with really sophisticated tools. It's like having this whole team of expert detectives all working to solve this big medical mystery.
And the more information they have, the better their chances of finding the right treatment for each patient. Let's zoom out for a sec, though, and look at the bigger picture of computational biology and bioinformatics. What other cool advancements are you seeing?
One area that's super interesting is how artificial intelligence, or AI, is being used to predict those new protein mutations in viruses. This is especially important with COVID-19 and other new infectious diseases that keep popping up.
Yeah, it seems like every time we turn around, there's another variant. It would be a total game changer if we could predict those mutations ahead of time. How are they using AI to do that?
Researchers are using this idea of a SARS-CoV-2 dialect. Basically, they're feeding these AI algorithms huge amounts of genetic data from all the different variants. And by analyzing these patterns, the AI can figure out the grammar of viral mutations. Then it can predict which changes are most likely to happen.
So it's like a crystal ball for viral evolution.
Yeah.
That's amazing. How does that help us in a practical way, though?
This predictive power could completely change how we develop vaccines and treatments. Imagine being able to design vaccines that target not just the current variants, but also the ones that are most likely to emerge in the future. We could actually stay ahead of the virus and maybe even prevent those big outbreaks.
That would be incredible. Completely change how we deal with infectious diseases.
Yeah.
Potentially save so many lives. Speaking of changing healthcare, I read this article about a really futuristic concept, self-driving laboratories.
Self-driving labs are basically these automated systems that can do experiments, analyze data, even optimize research protocols, all with very little human involvement. It's like having a whole team of robot scientists working 24/7.
That sounds amazing. What does that actually look like in real life?
Imagine a lab where robots handle all those dangerous chemicals, perform super precise experiments, and analyze huge amounts of data super fast. These labs could speed up research and discovery so much, lead to faster breakthroughs in medicine, material science, all sorts of fields.
Wow, that's mind-blowing. But what about us humans? Will we still have jobs in these futuristic labs?
Oh, definitely. Self-driving labs aren't about replacing scientists, but about helping them. By taking care of all the boring, repetitive tasks, these systems free up researchers to focus on the big questions, the kind that need human creativity and intuition.
That's a great example of how technology can help us be better, not replace us. Speaking of companies doing amazing things, we should talk about PacBio. Their president, Christian Henry, gave this great keynote address at the J.P. Morgan Healthcare Conference, where he talked about their vision for the future of genomics.
PacBio is really well known for its long-read sequencing technology. It can read much longer stretches of DNA than those traditional methods, giving us a more complete and accurate picture of the genome, especially in those complex areas that used to be difficult to figure out.
So it's like having a high-resolution map of this huge, intricate landscape. We can see all these hidden details that we missed before. What makes PacBio different from other genomics companies, though?
Well, while other companies are moving into things like proteomics and multiomics, PacBio is really sticking with genomics. They truly believe that genomics is the key to understanding human health and disease.
That's a bold statement. Why are they so sure about genomics?
Henry made a point of saying that genomics is the foundation for developing better diagnostics, treatments, and maybe even cures for lots of different conditions. They believe that if we can crack the code of our genes, we can create personalized medicine and improve healthcare for everyone.
It's a really inspiring vision, and based on all the amazing breakthroughs we talked about today, it seems attainable. Let's go back to AI for a minute. This article in Nature called Why AI Will Never Be Able to Acquire Human-Level Intelligence really caught my eye. It's like it's challenging the whole idea of AI taking over.
That article really goes deep into what makes human intelligence different from artificial intelligence. AI is great at handling tons of data and doing complex calculations, but it struggles with the things we take for granted: consciousness, creativity, emotional intelligence.
So maybe AI can imitate some parts of human intelligence, but it can't truly replicate the whole human mind. Does that mean it's all just hype?
Not at all. AI's strength is in how it can work with us, not replace us. Think of it as a powerful tool to help us analyze data, solve problems, and make smarter decisions.
It's like a super assistant that handles the hard stuff so we can focus on what makes us human. But even with all this incredible AI progress, we have to be careful.
Absolutely. AI can be misused. We have to make sure it's developed and used ethically with a focus on making life better for everyone. It's a huge responsibility.
It's a lot to think about, that's for sure. Finding that balance between the potential of AI and the responsibility that comes with it. But let's switch gears one last time and talk about another cool company in the genomics world, Oxford Nanopore.
Yeah, they're known for their portable, real-time sequencing tech. They can sequence DNA and RNA right there on the spot.
No more huge equipment and waiting forever for results.
Exactly. Their devices are tiny, they can fit in your hand and give you results in minutes.
Wow, that's like having a pocket-sized lab. What could you even do with that kind of tech?
Oh, so many things. Imagine tracking the spread of a disease outbreak in real time, or discovering new species in the middle of a rainforest. You could even diagnose genetic disorders right there at the doctor's office.
That's straight out of Star Trek. It could change everything. Healthcare, environmental monitoring, so many fields. What about Illumina? They're a big name in genomics too, right? What are they working on?
Illumina has been a leader in next-generation sequencing for a long time. They've made genome sequencing way more affordable. They're still innovating in that area, but they're also moving into things like proteomics and multiomics.
Proteomics is all about studying proteins, right? And multiomics is looking at all sorts of biological data together, like genomics, proteomics, and metabolomics. Why is that important?
Because it gives us a much more complete understanding of how things work. Instead of just looking at the genome, we can see how genes, proteins, and other molecules all interact with each other.
It's like going from a flat map to a 3D model. So as we wrap up this deep dive into genomics and bioinformatics, what are the main takeaways for our listeners?
This field is moving so fast. We have these groundbreaking technologies like RNA sequencing, AI algorithms, and portable sequencers. They're revolutionizing healthcare, making personalized medicine a reality, speeding up drug development, and helping us control diseases more effectively.
It's amazing to be living in this time, seeing how biology, technology, and human ingenuity are all coming together. But we can't forget about the ethical side of things.
Right. We have to make sure these advances benefit everyone, not just a few. And we need to be aware of the potential risks and try to prevent any unintended consequences.
It's a call to action for scientists, policymakers, and all of us to work together and make sure the future of genomics and bioinformatics is a good one. A future that's healthy, sustainable, and benefits all of humanity. Thanks for joining us on this deep dive. Keep exploring the world of science, and remember, knowledge is power, but knowing how to use it wisely is even more important.